Now that you've seen how DNA stores genetic information in that vault called the nucleus, and we use RNA to make proteins from that genetic information, which will help determine our traits, it's time to look at how those traits will be inherited. Gregor Mendel was the famous monk who first came up with some of the principles and understood the principles as far as how genes were inherited from parents. Even Darwin, who was about 20 years earlier, when he made his theory of natural selection, had no idea how things were inherited. Him and Lamarck, who we learn about in later units, knew that offspring were like their parents, but they didn't know how it got there. Well, Monk had done a whole bunch of studies with pea plants and was able to look at some traits based off of one gene and the patterns with which they were inherited. So here's one example. You have what is called the parent generation, and then F1 is for first filial and F2 for second filial, so those are like the next two generations. And he had yellow peas and there were green peas, and he bred those plants until they were true bred. Or sometimes you might, when you think of animals, they're called purebreds. And so the green ones only bred green plants, and the yellow ones only bred yellow plants no matter what. Then he took those two and he crossed them, and he found an interesting thing, that all the plants that were the offspring of those, all of them had yellow peas. Then he took those and bred them again and found out that about 3 out of every 4. And so this pattern was kind of curious, and it led him to some hypotheses as to how this works, and we'll need some terms to help identify and describe these different plants. So what he determined first of all is that there's more than one form of a gene, and they call these alleles. And in the case you just saw, the yellow peas were one allele, and the green peas would be the other allele that you can have. For each inherited trait, an organism has 2 alleles, and one comes from the mother and one from the father. And if those 2 alleles are the same, for instance, you have yellow and yellow, yellow from the mother and yellow from the father, we would refer to that as homozygous. Homo meaning the same, and then zygos referring to the zygote that is created when those 2 alleles combine from the parents. If they're different, for instance, a yellow and a green, that would be referred to as heterozygous, because hetero means different. If the alleles are different, like we saw in the example, we had 2 purebreds, so we had yellow and yellow. That would be homozygous if it was purebred, and then the other one was green and green. And when they bred, we had something that had a yellow allele and a green allele, so it's heterozygous, these top 2 being homozygous. The trait that is expressed is referred to as dominant, and the trait that is suppressed is referred to as recessive. So in this case, because that first generation was all yellow, yellow is dominant and green is recessive. He also came up with the law of segregation that says the sperm or egg carries only one allele for each inherited characteristic, basically saying this parent might have been yellow and yellow, but it can only pass on one allele to the offspring, which of course is going to be yellow. Now when you have a yellow-green in that first generation, it can pass on either the yellow or the green, but not both, because it can only pass on one allele to the next generation. So going back to the example, since yellow is dominant, we often use a capital letter to represent it, and green is recessive, we will use the same letter to show that they're coding for the same gene, even though they're different alleles, and the recessive one is usually lowercase. So the one parent, purebred, was homozygous yellow, big Y, big Y. The other parent was homozygous green, little y, and the offspring was heterozygous, big Y, little y. So this is kind of how this would look. You've got the 2 big Y's from the one parent, the 2 little y's from the other parent. Now, one of these, that law of segregation, only one of these can get passed on. So you have a big Y from that parent, and only one of these can get passed on. So you have the little y from the other parent. And that is why everybody in that first generation was heterozygous, because no matter which of these 2 you get passed on, they're both the big Y's. And no matter which of these 2 you get passed on, they're both the little y's. And so the P will be yellow in that first generation because yellow is dominant. So here's the list of alleles and we can call these alleles that you see, these letters are called the genotype, and the colors that go with them are the phenotype. And so the phenotype for the dominant homozygous is yellow, the phenotype for the recessive homozygous is green, and the phenotype for the heterozygous is yellow. And note, there is not a blending of traits here, and I think people used to think that for a long time, and there are some traits that work that way, and we'll talk about that in the next lesson, but most of the traits are dominant recessive. Now, where do these genes come from? Here is a picture, or a karyotype, of all the chromosomes on a male, and you can tell it's a male because you've got the stubby little X chromosome here and the big Y chromosome here, and we'll talk a little bit more about sex determination later. But the genes, you can see, are these little bands that match on the chromosomes. And on those little bands, there might be a different allele. You know, maybe this one has a big Y and this one has a little y. There could be the same alleles. Maybe both of these have, we'll use a P for instance, a little p. But those little bands are called locus, or plural is loci, and that's where the genes are found. So when we undergo meiosis, you remember one chromosome splits off, and they split off, and so that they each go to one gamete, so that you go from this diploid karyotype that we have in a gamete we're a haploid, and that's why one and exactly one of each gene gets passed on. So let's look at an example here. We've got a parent generation where you have an inflated pea pod and a constricted flea pod, and then the next generation are all inflated. So what does that tell us? Well, if these were pure to start with, then we know that all of these are heterozygous, and that inflated is dominant. So we could refer to inflated as a big I, and constricted as little i's if you wanted. And that means everybody in this generation is a big I, little I. And when the big I, little I crosses, you could get a big I and a big I. You could get a little I and a big I. And we'll always put the capital letters first. You could get a big I and a little I. Or you could get two little i's. And so 3 of the 4 will show that dominant inflated trait and will be constricted. The other thing you might have noticed is that when we combine these, we did first, then we did the inner, then we did the outer, and then we did the last. And you might remember that foil from math class, and it's the same way you could do genetics. So once again, this first generation is called the parent generation. They are all homozygous in this case, or true bred. Our second generation is called, or the offspring is called the first filial generation, F1 generation, and they were all heterozygous. And then our second filial generation, or F2 generation, we've got that combination where we can say the genotypic ratio is 1 to 2 to 1. There's 1 big I for every 2 big I, little i's for every 1 little I, or that the phenotypic ratio is 3 to 1. And once again, the genotype refers to the alleles, and the phenotype, let's go with that f sound, is the physical appearance. And that is our overview of genetics. And we'll get some more examples where we'll be using a lot of these words again, but if you need to re-watch this, feel free to do so. There's a lot of vocabulary, and we'll have a Quizlet so that if you want to just quiz yourself on some of those words as well, you can.